Myelination is the next process by which oligodendrocytes in the CNS and Schwann cells in the peripheral nervous system wrap axons with myelin, a lipid-rich sheath that insulates axons and speeds up action potential propagation. In the central nervous system, the white matter is about 99% myelinated, showing the vast importance of fast communication times for performing functions. Oligodendrocytes extend processes to multiple axons, wrapping them in concentric layers of myelin. Myelination in the CNS begins in the spinal cord and progresses rostrally to the brain, with regions such as the corpus callosum myelinating last. This process continues into early adulthood, with significant implications for cognitive development. Schwann cells myelinate individual axons in the peripheral nervous system. This process starts earlier than in the central nervous system and is crucial for the rapid conduction of nerve impulses in peripheral nerves. Here, a group of disorders known as leukodystrophies are characterized by defects in myelination in the CNS. Examples include Krabbe disease and adrenoleukodystrophy. These conditions often present with motor regression, cognitive decline, and seizures. It's notable to mention that multiple sclerosis is typically a disease of young adults. Early onset MS can occur and is characterized by immune-mediated demyelination. This condition can lead to a range of neurological deficits, including sensory disturbances, motor weaknesses, and epilepsy. Hypermyelination and other myelination disorders can also lead to epilepsy due to disrupted signal transmission in the brain. For example, Pelizaeus-Merzbacher disease, a hypermyelinating leukodystrophy, which is associated with spasticity, ataxia, and seizures. Now, looking at the development of the peripheral nervous system. The peripheral nervous system develops primarily from neural crest cells and placodes, which are specialized ectodermal regions. Neural crest cells are multipotent cells that migrate extensively throughout the embryo to form various components of the peripheral nervous system, including sensory neurons that form the dorsal root ganglia and transmit sensory information from the periphery to the CNS, autonomic neurons that develop into the sympathetic and parasympathetic ganglia, controlling involuntary functions, and Schwann cells that myelate peripheral nerves. Placodes contribute to the formation of cranial sensory ganglia, such as those for the cranial nerves, which are involved in spatial senses like vision, hearing, and balance. Notable congenital conditions of the peripheral nervous system include Charcot-Marie-Tooth disease, which is an inherited peripheral neuropathy caused by mutations affecting Schwann cells or the myelination of peripheral nerves. CMT leads to muscle weakness, atrophy, sensory loss, usually without direct CNS involvement, though severe forms can have CNS manifestations involving epilepsy. Also, congenital insensitivity to pain, which is a rare condition caused by mutations in genes involved in the development of nociceptive, which are pain-sensing neurons. And patients with this disorder do not feel pain, which can lead to repeated injuries and unnoticed infections. While PNS disorders primarily affect peripheral nerves, certain hereditary sensory and autonomic neuropathies can involve the CNS and have been associated with epilepsy. Programmed cell death. During development, the nervous system generates more neurons than are ultimately needed. Apoptosis selectively eliminates excess neurons to refine the nervous system. Neurons that fail to establish proper synaptic connections or receive insufficient trophic support, such as from nerve growth factor, undergo apoptosis. Rett syndrome, a neurodevelopmental disorder primarily affecting girls, is caused by a mutation in the MECP2 gene. It is involved in disrupted neuronal apoptosis, leading to microcephaly, intellectual disability, motor abnormalities, and epilepsy. Spinal muscular atrophy is caused by mutations in the SMN1 gene, leading to the loss of motor neurons through increased apoptosis. SMA presents with progressive muscle weakness and atrophy. While it primarily affects the spinal cord, some forms can be associated with seizures and epilepsy. Imbalances in apoptosis are linked to epilepsy in several ways. For example, excessive neuronal death in the hippocampus is a hallmark of temporal lobe epilepsy, where excitotoxicity and apoptosis contribute to the loss of inhibitory neurons and the development of chronic seizures. Finally, on the topic of postglial development and plasticity. After birth, the CNS continues to mature, with ongoing synaptogenesis, synaptic pruning, and myelination. Synaptic pruning eliminates excess synapses, refining neuronal circuits based on experience and activity, a process critical for learning and memory, known as critical periods, during which experiences have a profound impact on the development of specific functions such as vision and language that use experience-based plasticity. During this period of the postnatal brain undergoing periods of heightened plasticity known as critical periods, there are several associated conditions and links with epilepsy. Fragile X syndrome, the most common inherited, is caused of intellectual disability caused by mutation in the FMR1 gene. It leads to abnormal synaptic plasticity, resulting in cognitive deficits, behavioral issues, and high prevalence of epilepsy. Cerebral palsy, a group of disorders resulting from brain injury during perinatal period, affects motor control and posture. CP is often associated with epilepsy due to abnormal brain plasticity and connectivity. Epilepsy often arises from abnormal synaptic plasticity, where excessive or inappropriate synaptic connections lead to hyperexcitability. This is particularly evident in conditions like West syndrome of infantile spasms, where abnormal brain development and plasticity lead to early onset epilepsy and cognitive impairment. Thank you for tuning in on our video, and hopefully we have been able to outline to you the process of neurodevelopment, and especially focusing on the clinical link towards epilepsy, as epilepsy is really this hypersynchronous or excessive firing condition which is heavily linked to neurogenesis and synaptic plasticity that is crucial and is formed really in neurodevelopment, and why epilepsies usually have the strong genetic background and environmental influence for later parts of development after birth. So, thank you for watching. Now you know epilepsy, you know neurodevelopment to the T. So please follow us, like and subscribe, and we will have more videos coming out, whether it's on immunology in the field of medicine or other videos such as electromagnetism in the field of physics. Thank you, and we'll see you next time.